Okay, so welcome back to this next video in which we are discussing the protein kinase C pathway. Okay, so we've discussed now the production of these two products, diacylglycerol and inositol 1,4,5-trisphosphate from phosphatidylinosytol 4,5-bisphosphate, and this is done by the action of the phospholipase C beta enzyme, which has been activated by the alpha subunit of our GQ/11 heterotrimeric G protein. Okay, so we now want to discuss what these two products are going to do, basically. Now, we're going to start off with the inositol 1,4,5-trisphosphate, but we will come back to the diacylglycerol later. Okay, right. So, inositol 1,4,5-trisphosphate is going to go into the cytoplasm, and it's going to go to the membrane of an intracellular organelle, namely the endoplasmic reticulum. Okay, and it's going to act on receptors for inositol 1,4,5-trisphosphate, which sit within the ER membrane. Okay, and these are called IP3 receptors. So let's now have a look at the structure of these IP3 receptors. So they are calcium channels, so whenever drawing an ion channel, it's a good idea to start by drawing a cylinder. So here is the beginning of our IP3 receptor. Okay, and basically these IP3 receptors are tetramers. Okay, so they're made up of 4 separate proteins all stuck together to make the full ion channel. And I want to stress that this is the cytoplasmic side now of this membrane. Okay, so this faces into the cytoplasm, and this here, this is the ER lumen side. So this is within the intracellular organelle. Okay, right. Now, what I want to do is study the structure of one of these IP3 receptors in a little bit more detail. So I want to pull out one of these subunits for an IP3 receptor here. Okay, so I want to pull out this thing that I've highlighted in pink here and have a look at its membrane spanning topology in more detail. Okay, so again, I'm drawing the ER membrane here, which is again a phospholipid bilayer, and this side will represent the cytoplasmic side, and this side the ER lumen side. Okay, and basically the proteins that make up a quarter of an IP3 receptor, they have their amino termini in the cytoplasm. Okay, then they have the first membrane spanning alpha helix, the second, the third, the fourth, the fifth. Then they have a structure known as the P loop here, and then they have the 6th membrane spanning alpha helix, like so, and then they have their carboxylic acid terminus again in the cytoplasm of the cell. Okay, right. So these proteins that make up a quarter of an IP3 receptor, the subunits of the IP3 receptor, they are massive proteins. Okay, they have around 2,750 amino acids. Alpha alpha means amino acids. Okay, so they're big proteins. Now, there are 3 separate genes which can make up IP3 receptors. Okay, so the simple thing would be if there was just one gene that coded for the IP3 receptor subunit, but it's not that simple, basically. There are 3 known genes which code for different proteins which have different sequences of amino acids, but which all have this same membrane spanning topology, like so, and can tetramerize together to make a full IP3 receptor. Okay, so the IP3 receptor is a tetramer. Okay, and these 3 IP3 receptor subunits are known as the IP3R1 subunit, the IP3R2 subunit, and then also the IP3R3 subunit. Okay, right. So basically what we then need to ask is, how can you actually tetramerize these together? We have these 3 different subunits for IP3 receptors. Is it the case that you can only build homotetramers, where you use 4 of the exact same IP3 receptor subunits in all 4 of the slots? Or can you instead build heterotetramers, where you have different IP3 receptor subunits in the same IP3 receptor? Okay, so you do indeed find homotetramers, but we know also that you do find heterotetramers as well. So there are both homotetramers and heterotetrameric IP3 receptors. But overall, there are these 3 different subunits of IP3 receptors. Okay, right. So what we now want to discuss is how IP3 affects them, because it doesn't quite cause them to open, okay? It's more complicated than that. And this strange mechanism by which IP3 activates the IP3 receptors is responsible for the different types of calcium signal that you can actually produce within a cell, which I'll discuss with you in a moment. Okay, right. So, let's start with what IP3 is going to do to the IP3 receptors. So what I'm going to draw is I'm going to draw an IP3 receptor from above. Okay? So we're now looking down from the cytoplasm at the top of an IP3 receptor. Like so. So here is the pore through which calcium can move. So remember, the ER is a good store of calcium within the cell. Okay, I suppose I should have just revised what the endoplasmic reticulum is, so we'll do that quickly. So the endoplasmic reticulum is an intracellular organelle, okay, shown here. So it's a membrane-bound structure within the cytoplasm of the cell. Now, it's a good store of calcium. Okay, so calcium ions are very low within the cytoplasm. The concentration of calcium intracellularly is around 100 nanomolar. Contrast that to the concentration extracellularly, which is around 1.5 millimolar. That's a 15,000... Oh, whoops, sorry, you can't see this. That's a 15,000-fold gradient across the cell membrane. However, we do have a store of calcium within the endoplasmic reticulum, and we can release this calcium into the cytoplasm, and it has important signaling properties. Okay, and that's what's going to happen when we signal with IP3. Okay, so here is our IP3 receptor. Now, initially, on the extracellular aspect, you have binding sites for calcium exposed. So these in orange, these are representing binding sites for calcium. And these are inhibitory calcium binding sites. Okay, so if calcium binds to these inhibitory calcium binding sites, the likelihood that the IP3 receptor will open will be decreased even further, basically. So these are inhibitory calcium binding sites. Okay. Now, you also have IP3 binding sites, which I'll show in blue here. So these sites in blue, these will represent the IP3 binding sites. Now, at present, there is no IP3 bound to these IP3 binding sites. Now, notice there are 4 IP3 binding sites because there are 4 subunits within the IP3 receptor. Okay, right. So when IP3 goes up, because we're producing it from phosphatidylinositol 4,5-bisphosphate breakdown, the IP3 will bind to these IP3 binding sites on the IP3 receptors. Okay, so you'll get 4 IP3 molecules binding, 1 to each of the binding sites. And then what it triggers is it doesn't trigger the channel to open. Instead, what's going to happen is all of the subunits are going to change conformation, and they're going to take away the inhibitory calcium binding site. Okay, so the inhibitory calcium binding site is going to be buried underneath other portions of the protein so that it's no longer exposed to the cytoplasm. Okay, and instead you're going to make available a new calcium binding site, which I'll show in green over here, which is a stimulatory calcium binding site. And if calcium now binds to that stimulatory calcium binding site, it will cause the IP3 receptor to open. Okay, so this is the stimulatory calcium binding site. Right, and I'll also show the IP3 binding sites on this second picture as well. Okay, so these are stimulatory calcium binding sites, so I suppose I should have multiple arrows here. Okay, since I've used the plural. Now, I'll also show the IP3 receptor, the IP3 binding sites on each of these subunits. Okay, so what happens then, to review this, is when IP3 binds to these IP3 binding sites on the cytoplasmic side of the IP3 receptor subunits, what will happen is they will all change conformation so that they take away this inhibitory calcium binding site and put up a new stimulatory calcium binding site. Now, if calcium comes and binds to the stimulatory calcium binding site, it will trigger this IP3 receptor to open and it will release calcium. Okay, so IP3 receptors actually don't just require IP3 to open them. They require IP3 to put them into a state where then calcium can come and open them. Okay, now it's still a little bit of a mystery where the initial calcium that opens an IP3 receptor comes from. Okay, it's believed that it must come from the extracellular fluid. Okay, so some calcium channel must open that then allows a little bit of calcium to come in, which then binds to the stimulatory calcium binding sites on these IP3 loaded IP3 receptors, and then causes them to open, and then they release a large amount of calcium from the ER. So let's explore this more now by looking at the different types of calcium signal that you can get within a cell. Okay, right, so I want to discuss calcium puffs and calcium waves. Okay, and these will result from different levels of stimulation of the cell. Okay, so let's draw our cell here, and I'm going to elongate it because this works better if we have an elongated cell like so. Okay, and we'll have the ER here. Now, let's start with a calcium puff. Okay, so a calcium puff is going to occur when you only stimulate part of the cell with your initial ligand. Okay, so this little portion of the cell membrane that I'm now colouring in purple here, this portion is going to have the initial ligand put onto it for that G protein coupled receptor. So the ligand is going to bathe only this portion of the cell membrane, not all the other portions of the cell membrane. Okay, so it's a local signal. Okay, so it doesn't signal to the entire cell membrane. That would be a global signal. We'll discuss what happens then in a moment. Now, local signals are also often short signals. Okay, so the ligand will come and then go very rapidly. So it's a local and short signal. So for instance, neurotransmission will be an example of this sort of a signal. Okay, right. So what will happen is the G protein coupled receptors in this region will become active, which will result in the phospholipase C enzymes in this portion of the membrane becoming active and then producing IP3. Okay, so in this portion of the cytoplasm here then, you will get a rise in IP3 levels. Okay, so IP3 is going to go up in this portion here. And that means that the IP3 receptors in this portion of the ER here are going to be exposed to IP3. Okay, so this portion that I'm colouring in green, this is where the IP3 is going up, whereas the portions over here will not be exposed to IP3. Okay, let's now evaluate what this is going to mean. So if I draw a bigger picture of that ER here, okay, so this represents the endoplasmic reticulum within our cell, then we've got IP3 receptors all the way through this ER. Okay, so let's draw some of these. So here's a second one, here's a third one here, and I think we'll draw 5 to get the message across. So here is a 5th one over here. Okay, right, and then we'll, oh sorry, that's not the 5th one, that's the 4th one. And then we'll have a 5th one over here. Okay, right, so these 3 in the middle here, and of course this is oversimplified, in reality there'll be millions in this little green patch here. Okay, but for our picture we've only got 3. Okay, so these 3 will be bathed in IP3, these 2 will not, which means that these ones will go into the state where calcium can now activate them. Okay, whereas these ones will not. Now, something then allows one of these to get some calcium, basically, and that part still isn't understood. We don't understand where the initial calcium comes from, but we think it must come from the extracellular fluid through some sort of ion channel in the cell membrane. So it's likely that some sort of ion channel in the cell membrane allows a little bit of calcium to come in. This will then go and activate one of these IP3 receptors here, or multiple potentially. Okay, but a few of them is the point. So let's say it activates this one here in the middle. Okay, so calcium now binds to this, and it's already had IP3 bind to it, so it has stimulatory calcium binding sites exposed. So now it's going to be activated to open. Okay, and when it opens, it will release calcium from the lumen of the ER out into the cytoplasm. The calcium will now spill over onto its neighbours, okay, and those have been bathed in IP3 as well. So they've had 4 IP3 molecules bind. They now have 4 stimulatory calcium binding sites exposed, and the calcium that's spilling over from this one will now activate them to open, so you'll get the release of calcium from these two as well. Okay, like so. So out comes more calcium. Now what will happen is calcium will spill over onto these two, but they haven't had IP3 bind to them. Okay, so they do not have stimulatory calcium binding sites exposed. So they will have calcium bind to their inhibitory calcium binding sites, and they will absolutely not open. So they do not propagate this calcium signal. So what you'll get is calcium going up around these IP3 receptors which have received IP3 basically. So you'll get a calcium signal that will look like this, i.e. you'll get a local calcium signal. Okay, right, what will then happen is the ligand was only delivered for a certain amount of time, okay, and then it's going to be broken down, let's say. So the G protein coupled receptors will become inactive, the heterotrimeric G proteins will inactivate themselves, and now because the G protein coupled receptors are inactive, they won't reactivate the heterotrimeric G proteins. Therefore, phospholipase C will turn off, okay, so you'll get no more IP3 being produced, and the IP3 that you have produced will be gradually broken down. Okay, so IP3 is now gone, let's say, and the IP3 receptors have lost their IP3, and in addition, the calcium is going up in the cytoplasm around them. Okay, so what will happen is when calcium gets to a too high level, even if the IP3 receptors have still got their IP3 bound to them, which let's assume they still have at the moment, okay? So let's assume the IP3 hasn't gone down that quickly that they've lost it yet, okay? So even if they've still got their IP3 bound to them, once calcium gets too high in the environment around them, calcium goes from having a stimulatory effect on them to inhibiting them, okay? So the too high calcium level causes them to shut down. So even when they've got IP3 bound to them, low levels of calcium will activate them by binding to those stimulatory calcium binding sites. But if you put really high levels of calcium, that will inhibit them, okay? So when they've released too much calcium, they'll start to be inhibited, and then they'll close, and whilst they're closed, the IP3 will go down, they'll lose their IP3, and then we're back to square one. Okay, and then the calcium will gradually be pumped back into the ER lumen, okay, by a pump known as SERCA. So let me just introduce this pump here. So there is a pump on the ER membrane called SERCA, which stands for the sarco, that's the S, / endoplasmic, that's the E, and then the R is for reticulum, okay, and then the Ca is for calcium ATPase, okay, and basically the SERCA is an active transport mechanism for returning calcium into the ER. It moves 2 calcium ions into the lumen of the ER in return for 3 protons being moved out of the lumen of the ER. Okay, and every time it does that, it will also hydrolyze a molecule of adenosine triphosphate to adenosine diphosphate and an inorganic phosphate molecule. Okay, right. So what will happen then is once the IP3 receptors have stopped releasing calcium and they're gradually losing their IP3, the Circa pumps will then return the calcium into the ER, and then the calcium signal will vanish. So this local signal in calcium that you get, which happens for a short period of time, this is known as a calcium puff. Okay, we're going to now contrast that to another type of calcium signal that can be generated, which is called a calcium wave. Okay, and this results from a global stimulation of the cell by that ligand. Okay, and generally global signals are also prolonged signals. Okay, so what we're imagining now is if this is our cell here with our ER in, the entire membrane is going to be bathed in the ligand, basically. Okay, so I'll just label this up as the ER. So we're going to put the entire membrane in the ligand. So I'll colour in the entire membrane in purple here. And this represents bathing the entire cell in the ligand. Okay, and what will happen is the ligand will activate the G protein coupled receptors, which are all over the cell membrane. That will then activate phospholipase C of the beta form, and you'll be getting IP3 being produced all over the place. Okay, so you're getting a global rise in IP3, so this is a global signal, and we're also going to say that it's a prolonged signal. Okay, so we're not going to just have a transient signal here. We're going to leave the ligand on for a long period of time, so we're going to be producing IP3 all the time. Now you might think, well, will IP3 just get higher and higher and higher within the cytoplasm? No, eventually it will get to an equilibrium where the amount of IP3 being broken down is the same as the amount that's being produced, but that will be at a significantly high concentration of IP3 in the cytoplasm. So the point is that we've got IP3 at a very high level all over the place within the cytoplasm. Now, what this produces is repetitive calcium waves that propagate along the cell. Okay, so let me explain what is understood about these calcium waves, and there are still some massive questions which are not known. Okay, right. So basically, what's going to happen this time in our ER, okay, so if we draw our ER again, is that the IP3 receptors all the way along the ER are going to have IP3 bound to them. Okay, so absolutely every IP3 receptor will have IP3 bound to it. Okay, and therefore it will be in the state where low levels of calcium can activate it to open. However, I want to stress again that if you have extremely high levels of calcium, that will not activate it to open, that will cause it to close, okay, and that's going to be important. Then, something initiates the whole chain reaction off, okay, and again, it's not understood what this is, but we think calcium must come in through an extracellular route, basically. So through some ion channel on the cell membrane, calcium is going to enter from the extracellular fluid coming into the cytoplasm. It will then activate the first IP3 receptor, which let's say is this one here, just so that we can have a beautiful wave. Okay, this one will release calcium now from the ER. Once the calcium from the extracellular fluid activates it, it will open, release calcium out. The calcium will then overflow onto its neighbour. The calcium ions will activate this IP3 primed IP3 receptor. So it will now release calcium. Okay, calcium from it will overflow onto its neighbour, and these will bind to those calcium stimulatory calcium binding sites, and it will open and release calcium. Okay, and the process will continue on propagating through the entire cell like so. Okay, so you're getting this propagating release of calcium from the ER. Now, whilst the release of calcium is propagating, you also have to factor in the fact that calcium will be building up over here. Okay, so calcium will build up over here, and what that will do is it will feed back on the IP3 receptor and cause it to close. Okay, so what's going to happen is whilst the wave of release is propagating forwards, behind it these IP3 receptors will start to close, okay, because of the high calcium level. And even though they have IP3 bound to them, they will close in the presence of too high calcium levels. So this will close, and then what will happen is the calcium will be returned back into the ER membrane by our Circa pump, okay? Which, remember, will pump 2 calcium ions in for 3 protons out, and also the hydrolysis of ATP. So calcium will now fall down here, okay, and then the falling calcium will also propagate along. So you're getting calcium rising here, causing calcium rising here, causing calcium rising here, and of course, as it propagates forward, behind it, the calcium will be going down. So basically, what you're going to get is something that looks like this if we plot a graph. Okay, right, so if we have time on the x-axis here, and that's on the y-axis have calcium concentration. Okay, if we plot this at place 1, position 1 over here, what will happen is calcium will rise and then it will fall, like so. Okay, so this is position 1. So let's colour it in a certain colour. Okay, so in green here, and this is position 1 here. Then, if we do it for position 2 here, position 2 will be activated to go up later than position 1. Okay, so it will be going up like so. Okay, and then it too will fall down. So once the calcium gets to a too high level, it will cause the IP3 receptors to close, and then the Circa pumps will return the calcium into the ER. And then, of course, if we do position 3 over here in blue, this will be slightly delayed of position 2, basically, because it's taken longer for the propagation of the release to reach position 3. So it will be starting to go up just as position 2 is starting to go down. Okay, so that's basically the phenomenon of a calcium wave, that the calcium signal propagates along the cell. You get a calcium spike here, which triggers a calcium spike here, which triggers a calcium spike here, in this saltatory conduction manner, basically. Okay, so it's just like the way an action potential propagates along, basically, where an action potential in one place triggers an action potential in another, triggers an action potential in the next, okay? And that phenomenon of a signal like that being carried by the signal here causing a signal there, causing a signal there, causing a disturbance there, causing a disturbance there, etc. This is called saltatory conduction. Okay, so saltatory conduction doesn't just refer to action potentials. It refers to the idea that a disturbance here can cause a disturbance there, 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 and that's how we can propagate this signal forward. Now, one more thing that is not understood at all is that you go round again, basically. You don't just fire one calcium wave, you then trigger another calcium wave, and another, and another, and another. So the cell, as long as it maintains the global signal, will continue to fire calcium waves like so. Okay? And the other the curious thing that really is not understood is that the frequency with which it fires calcium waves, so how long it waits between calcium waves to then fire another one, that is related to the IP3 concentration, okay, and therefore to the stimulus intensity. So let me explain that in more detail. If you put a higher concentration of ligand onto the cell in this manner, okay, so you bathe the entire cell in the ligand, then the frequency of calcium waves that you will get will be greater, okay, so you'll get them like this, basically. Whereas if you put a very low concentration, okay, but you bathe the entire cell, so you give it a global prolonged signal, you'll get much lower frequency calcium waves, okay. So basically, the stimulus intensity is encoded by the frequency of the calcium waves rather than the amplitude. You can't change the amplitude of the calcium spikes, okay? Instead, what you can change is the frequency of the calcium waves, okay? And this again bears similarities with action potentials. In action potentials, you can't change the intensity of an action potential. Instead, stimulus intensity has to be encoded in the frequency with which you fire action potentials. Similarly, In the case of calcium waves, stimulus intensity is encoded in the frequency of calcium waves rather than in the size of them. Okay, so those are the two different types of calcium signals which you can evoke within cells, depending on whether your signal is a local short-lived signal or a global long-lived signal. Okay, right, but the overall importance for us is that you're getting calcium going up in the you're getting calcium going up in the cytoplasm. Okay, and in the next video what we'll talk about is how this calcium going up in the cytoplasm, along with the diacylglycerol molecules, is going to lead to the activation of protein kinase C enzymes.